Finish chapter 6 or not. Get to there. Got to there. Before we begin, are there any questions? Can somebody please confirm you can hear me?
We can hear you.
Thank you. All right, you should be able to see my screen now. And close that down. All right, today is November 4th. Boy, that means the election's tomorrow. I've got to vote. Um, and we're supposed to be finishing chapter 6. I don't know if we'll get there, but and actually starting chapter 8 today. Any questions about any of that? Let me remind you the quiz will be next week. There was someplace, not on the web page, the Canvas web page, but I think in the grade book it had the quiz on the wrong date. I've now corrected that. Any questions about anything? Let me remind you, your infectious disease project is due this Saturday. I'll talk about that in the lab. All right, any questions? No, let's begin with chapter 6. I have that we had talked about the effect oxygen has on microbial growth when you're growing the bacteria or other microbe inside a thioglycolate tube. And when you first take the thioglycolate out of the autoclave, there will be no oxygen in the media because the media removes oxygen. However, oxygen moves into the media, excuse me, so that at the top of the media, there's complete oxygen, but at the bottom of the media, there'll be no oxygen. And that sets up an oxygen gradient in the tube. As you go down the tube, there will be less and less oxygen until at the end of the bottom of the tube, there's no oxygen. And you can see obligate aerobes only grow at the bottom of the tube, less growth as you go up the tube because there's more oxygen. And then obligate aerobes only grow at the top of the tube. And as you go down the tube, there'll be less and less growth because there'll be less and less oxygen. Any question about any of that? When we're talking about media, you should understand that media is a plural. When you're talking about one medium, you say the word medium. A media is a liquid or a solid solution containing all of the nutrients needed for microbes to grow in. You usually get it sterile, and in the microbiology lab, how we sterilize things is we stick them in an autoclave. A sterile media or anything that's sterile contains no living microbes, including endospores. An inoculum are the microbes you introduce into the media. The culture is then what we grow, containing both the media and the inoculum, meaning what you inoculated into the media to grow. We have a very wide variety of media available in microbiology. I'm not sure if I mentioned it before, so let me mention it now. If you were to come into the lab, I would pull out a book that's about this thick. It's actually a little bit wider or longer, and just a little bit longer than this, but a really big book. And in each page of the book, it discusses one medium. So it tells you how many different media we have in microbiology for you to use. Any question about any of that? Microbiological media contains the growth ingredients for microbes to grow, including an energy source, a carbon source, a source of nitrogen, sulfur, and phosphorus, a source of trace elements, and organic growth factors if an organism needs them. Like Streptococcus pyogenes would require all of these because it does need organic growth factors or it's not going to grow. But what about E. coli? Would you need organic growth factors in that one? Would E. coli grow without adding organic growth factors in the media? Nobody knows. E. coli would grow fine. It can make its organic growth factors. But what about trace elements? Does E. coli need trace elements? Yes, E. coli needs trace elements, but usually you get trace elements from either like the cow that you grind up and put in the media, or the beef extract, or maybe from the water itself. Supplies all the trace elements that would be needed. How about if we're growing a photosynthetic organism and give it light, and this organism can pull nitrogen out of the air to get its own nitrogen source? What would you need for that organism? Would you even need an energy source?
Yes.
Uh, they do need an energy source, but we're already supplying it by light because this is a photosynthetic organism. So it doesn't need any other energy source. How about a carbon source? Would a photosynthetic organism require a carbon source? That's sort of a trick question, but you can answer it either way. Nobody wants to guess after the last person got sort of wrong. Uh, most photosynthetic organisms can pull carbon dioxide down from the air or from the water if it's dissolved in the water. So most photosynthetic organisms would not need a carbon source. There are a few exceptions, and we're not going to talk about those exceptions. I already mentioned that this organism can pull nitrogen down from the air, so this organism does not need a nitrogen source, but it would need a sulfur source and a phosphorus source, as well as trace elements, which are usually in the water. Wouldn't need organic growth factors because photosynthetic organisms can make all of the growth factors that they need. Any question about any of that? All right. Agar is a complex polysaccharide derived from seaweed. It's used as a solidifying agent for culture media, and it's the number one solidifying agent, although you could have gelatin or starch. There might be a few other solidifying agent as well, but those are the main ones. But agar is by far the most widely used. Agar is usually in a petri dish because if you had a liquid broth in a petri dish, it'd just be too easy to spill. But you can put agar in a tube. We have two types of agar tubes. Let me see if I can get this. I think it's here. Oh, there it is. We have a slant where the agar and the media is at an angle like this, meaning we pour in liquid agar, meaning hot agar, and then we put the tube at a slant, tilting it, and then this would go straight across because the tube is slanted. And then you allow it to solidify, and then you have the slant like that. And then we have a deep. That's just where you pour agar into a tube and it goes across straight, sort of straight. Any question about any of those? You can also have broth in a tube, in which case it goes across straight, although it's not solid, it's a liquid. Agar is the most common solidifying agent used in microbiology because generally it is not metabolized by microbes. In fact, the reason why Dr. Koch first started using agar was because he was using gelatin. Before that, he was using, I think he was using a potato to grow his microbes. And you can imagine streaking on a potato was not very easy. I don't know if it's a cooked potato or a raw potato, but either way, it didn't work very well. And then he did it on gelatin. The trouble with gelatin is a lot of microbes can digest the gelatin, and then in which case you have a semi-solid or a liquid mess where all the colonies then mix together. And so Koch was complaining about growing his microbes on gelatin, and it was actually uh, the wife of one of his assistants in the lab who told him, well, maybe you should try agar. And agar is something that is used as a solidifying agent in cooking and baking. And he did, and he liked it because the microbes didn't digest it. And it's so well used that we still use it today as a solidifying agent. We have different types of culture media. We have a chemically defined media where the exact chemical composition of the media is known, meaning all of the additives to the media are things that you add to it. Let me blow that up a little bit. So here's a fairly simple media for growing things like E. coli. You put 5 grams of glucose, 1 gram of ammonium phosphate, 5 grams of sodium chloride, 0.2 grams of magnesium sulfate to be both a magnesium source and a sulfur source, and then potassium phosphate to supply potassium, which you usually get from like the water or something like that, and then phosphate, and then you add it to 1 liter water. And then that's all that's in this media. This is called the glucose salts media. You don't need to know that, but and that's all you need for things like E. coli to grow. This is a chemically defined media for a fastidious organism that has a lot of additives to it. Let me see if I can blow that up so I can read it. I'm not going to read all of them. I can't blow it up. Nope, didn't mean to do that. So it has glucose, starch, sodium acetate, sodium citrate, oxalic acid, a bunch of salts, sodium chloride, you know that one is a salt, but ammonium chloride, potassium chloride, calcium chloride, ferrous nitrate to provide both nitrate and then iron, a bunch of amino acids because Neisseria gonorrhea requires these, like cysteine, asparagine, glutamic acid, arginine, cysteine, a bunch of organic growth factors. There's NADH, uracil, biotin, a B vitamin. I don't know that one. And then this is sodium thioglycolate to help remove oxygen from the media. And then you add that to 1 liter of water. So this you would be supplying for a fastidious organism, and you can see you have a whole bunch of additives to grow in them. Chemically defined media are not usually used for fastidious organisms because you have to add so many things. Chemically defined mediums are usually only used for autotrophs, organisms that can make all of their own ingredients and then pull CO2 down from the air or the water. For most microbes, we have a complex media, which contains extracts or digestive yeast, meat, plants, providing vitamins, organic growth factors. The exact chemical composition of a complex media is not known because let's take a look at this one. A very simple complex media, nutrient agar. having peptone. This is a partially digested protein obtained from soybeans. Beef extract, so you grind up 3 grams of cow and put it in the media. Sodium chloride, and then agar, and that's all that is in this media. But we don't know the exact chemical composition because we don't know what the cow was eating and what molecules came from the cow. Any question about any of that? As well as, what molecules were in the protein digest from the soybean. Also, we don't know all the ingredients in that either. Peptone, a partial protein digest, is the primary energy source, carbon source, and nitrogen source, and sulfur source for many media like this one. It has more protein in 5 grams of peptone than in 3 grams of beef, why I'm saying the primary carbon and nitrogen source. But obviously, beef is supplying energy, carbon, and nitrogen as well. Complex media are usually used to grow heterotrophs, bacteria, and fungi that do require an organic carbon source, meaning they cannot pull carbon down from the air or from the water and CO2. Any questions about any of that? So what we would use this complex media for growing is things like E. coli, things we normally grow in the microbiology lab. That doesn't mean that E. coli requires it. As I mentioned earlier in the previous slide, this glucose salts media is all you need for E. coli to grow. And then glucose is providing the energy source and the carbon source. And that's all E. coli needs. But usually we grow E. coli in something like nutrient agar. We do have different types of media. We have an anaerobic media, and this is important for obligate anaerobes that must avoid exposure to oxygen. Anaerobic media have some reducing agent in it, such as sodium thioglycolate, that combines with and removes oxygen from the media. The result being oxygen depletion, at least at the bottom of the tube. You usually heat an anaerobic media to drive off the dissolved oxygen in the media, and then you rely on the reducing agent to remove oxygen, at least from the bottom of the tube. Oops, not one. We have selective media, and this is media that halts or inhibits the growth of a particular group of microorganisms, but not affecting the growth of other microbes. An example of a selective media is a media containing 7% sodium chloride. And we actually have a media called 7% sodium chloride. It has 7% sodium chloride in it, which is way too much sodium chloride for most microbes. Most microbes will not grow above about 2% sodium chloride. But this media has 7% sodium chloride, so it will select for salt lovers. We have a name for that. It's a halogen, halophile, something like that. Salt lover would exactly be halophile. An example of a bacteria that would grow on 7% sodium chloride media would be Staphylococcus aureus. And as you know, Staphylococcus aureus grows on your skin, and your skin can get up to 20% sodium chloride. As I mentioned, Staphylococcus aureus is not an obligate halophile because it can grow on regular media like nutrient agar, which is about, it's isotonic, so about 0.9% sodium chloride. But it is a facultative halophile because it can grow in 7% sodium chloride. In fact, it can grow higher than that. It can grow in at least 20% sodium chloride. Any question about any of that? We have other types of media. There's a differential media, and these media provide differences that can be usually visually distinguished or visually seen between different microbes. The differences are not related to how well the organism grows on the medium, but it makes it easy to distinguish colonies of different microbes. They may have a different color or they do something different in the media. Like, for example, a blood agar plate is a differential media. Let me blow this up. Because you can differentiate between the hemolytic activity of the bacteria. You have some bacteria which are alpha hemolytic, or that means no hemolysis, or gamma hemolysis. That's the lower Greek letter for a gamma, by the way. And you can see the blood are not impacted all the way up from the agar, all the way to the growth of the bacteria. Gamma hemolysis. We also have alpha hemolysis, which is partial lysis of the red blood cells. And the off-white color of the alpha is our bacteria. And then you can see a darkening of the agar around the alpha, as well as inside the alpha. And that is from the partial hemolysis of the red blood cells, making the agar look dark. Sometimes the partial hemolysis actually makes the agar look green, depending on the light you're using and some of the other characteristics. Anyways, that's gamma hemolysis and alpha hemolysis. And then this is beta hemolysis. The beta in white is the beta hemolytic organism, and it completely lyses the red blood cells. Why we see a clear zone around the beta and inside the beta from the complete hydrolysis of the red blood cells. You can actually hold this plate up to the light and see through the clear region where the beta hemolysis is. So that's why blood agar is a differential media, allows you to differentiate between the different hemolytic activity of different species of bacteria. Gamma hemolysis, alpha hemolysis, and beta hemolysis. And if you're wondering, you can actually see a ring of alpha hemolysis around the beta hemolysis. And that's normal. Any question about any of that? Another media we have are selective and differential media. These are media that are both selective and differential. An example of a selective and differential media is EMB, or eosine methylene blue. It selects against gram-positive bacteria because eosine and methylene blue are actually dyes which get in the cell wall of the gram-positive bacteria and prevent them from growing. I think they also have maybe bile salt in it, which also prevents the growth of the bacteria. I'd have to look that up, so don't quote me on that one. I don't remember if it's an EMB or not. But definitely it selects against gram-positive bacteria, but the gram-negative bacteria can grow on EMB plates. And so if you get growth, it's most likely to be a gram-negative, because the gram-positives are selected against. This is also differential on the basis of lactose fermentation. If you get the sugar lactose to be fermented, you'll see either dark colonies or there's some metallic green colonies. And that's from the buildup of acids from the fermentation of the sugar lactose. Meaning you ferment the sugar lactose, you make acidic products, making the pH indicator turn darkish or black or metallic green. If the bacteria, on the other hand, cannot ferment the sugar lactose, they will be the bacteria's normal color. In this case, we have white colonies on gram-negative bacteria that does not ferment the sugar lactose. Any question about any of that? We also have an enrichment media. This media encourages the growth of some bacteria, but inhibits the growth of other bacteria. Therefore, all enrichment media are selective. But an enrichment media is used differently because we use enrichment media to encourage the growth of a microbe that we want, like from a mixed culture. And let's say you're sampling bacteria from the soil, like you want to find a phenol digesting bacteria in the soil. Well, they exist, but their numbers would be very low. And what you can do is put that soil inoculant into an enrichment media and then start selecting for the phenol degrading bacteria. That will increase the numbers of the desired microbe to detectable levels. Like the phenol digesting bacteria, let's say it's 1 in 1 million cells, you'd have to streak out 1 million colonies before you would have a chance of getting one colony that actually digested phenol. So it wouldn't be practical. But what you could do is put it in an enrichment media. Let me stop the slide here. See if I have it next. Nope. So we have an enrichment media. And let's say the only carbon source in this media is phenol. That will select for microbes that can digest phenol, but select against other microbes. Now let's say its amplification is 100-fold. So we had 1 in 1 million. Let me see if I can get that. So 1 in 1 million is the cell of interest. Trying to get that out would not be very good. Well, let's say our enrichment media enriches by 100-fold. We now will have let's go over here. After enrichment, we'll now have 1 in 10⁴. Oop, I don't need that right. So you have one cell out of 10⁴. That still is not good enough to streak and try and get. You'd have to streak out 10,000 colonies before you get the one of interest. But what you could do is put another, just a minute here, let me get my brush back. You could put it in, oh, it's too thin. Put it in another enrichment media, and let's say it enriches once again 1 in 100. That would increase the number of microbes. It's not right, exactly right, but it's close enough. That will make it so that the bacteria of interest are 1 in 100, which is actually good enough. It is possible to streak out 100 different colonies and you would get one, which isn't a real good number, but my point is it is possible to do. You could also enrich it further, and we've already got it down to 1 to 100, so we'll just say it increases the enrichment. Any questions about how you use an enrichment media? And how you do this one? Let me get up here. And you just take an aliquot out, like 1 mil, put it into 9 mils. Any question about enrichment media? Enrichment media are often used when we're looking for a specific soil microbe or other environmental microbe, or when we're looking at a fecal sample and you want to know the bacteria that is causing the patient's illness. The chances are very great that the normal microbe of the intestines will be outnumbering the microbe causing the problem, in which case you may need to put the fecal sample in an enrichment media looking for a human pathogen in the fecal sample. I already talked about that. All right, this is the different types of culture media, chemically defined, a complex media, a reducing media, a selective media, a differential media, an enrichment media. And remember, you can have combinations like selective and differential. Obtaining pure cultures. A pure culture is a culture that contains one and only one species. Louis Pasteur first developed a technique for getting a pure culture. It was a very difficult way of doing it. He had to um, make many different dilutions, growing as microbes, as a mixed culture in liquid, and make many different dilutions, and then make 10 replicas of each dilution. And if he found one dilution where only one tube out of 10 tubes had growth in it, and the other 9 tubes did not have growth in it, Louis Pasteur correctly surmised that the one tube out of 10 that had growth in it was likely to be a pure culture. That's not an easy way of getting a culture, a pure culture. A much easier way of getting a pure culture was developed by Dr. Koch. And what he did was he streaked out the mixed cells on an agar plate to get isolated colonies. And then you take cells from the first colony plate and then streak it out to get isolated colonies on a second plate. And the cells that went through 2 colony isolation events are now greater than 95% chance that is a pure culture, which is good enough to use in microbiology. When we're counting the colonies on a plate, we usually call them not colonies on the plate to get our number of cells in original solution. We'll call them colony forming units, and then we'll say how many colony forming units were in the original solution. The reason why we call it colony forming unit is because a colony could be obtained from one individual cell, but at least 30% of the colonies on a plate are formed from 2 or more cells, including from a clump of cells. And that's why when you streak out your first plate for getting isolated colonies, you can't pull from one colony and expect that it will be a pure culture, because the chance of it being a pure culture is only 70%, meaning there's a 30% chance that that colony there is from mixed parentage, meaning mixed cells, 2 or more species. And 30% is just too high in microbiology. We want it to be 5% or less. And so what you do is you take from that isolated colony, streak it out on another plate to get isolated colonies, and then you take from one colony to set up your stock culture. When you have a stock culture set up after the cells have gone through 2 colony isolation events, the chance that you have a pure culture is greater than 95%. Any question about any of that? And that's why we call it a colony forming unit. And then when you say how many cells are in the mill of the original, You don't say how many cells per mil, you say how many colony forming units there are in the original solution. All right, streaking for colony isolation is shown here. Let me blow this up. What you want to do is streak out from your stock culture and streak it on sector 1 or quadrant 1. The highest concentration will be at the start of your streak. The lower concentration will be at the end of your streaking. Because as you streak, more and more cells will be added to the plate so that you'll be putting less and less cells down on the plate as you streak sector 1. You then sterilize your loop to kill everything that's on it, cool it, usually by touching it to a piece of the plate auger to cool it, where you're not streaking, like right here would be a good place. And then start streak 2, and as you go in streak 2, you go into streak 1, meaning sector 1, to pick up cells from sector 1, and then streak them out in sector 2. It's important that when you go into sector 1, you only do it about 3 or 4 times. And you do it from the region where sector 1 is at low density, meaning, come on, mouse, this region here. You do not put it in sector 1 where there is high density, this sector here. And then you streak out on sector 2. Oops. You then sterilize the loop, and then you cool it on the auger plate, and then you start streaking 3. It's not shown here, but you actually are streaking here, picking up the cells, and you want to do that 3 or 4 times. You don't want to do it just 1 time. Once again, you do not come into the region of sector 2 where the bacteria are heaviest. You come into the region where the bacteria are lightest. And then you streak out sector 3. Let me mention that when you're doing the sector streaking, you should have 20 to 30 streaks in each sector. That's not shown here because it would be too many lines for you guys to follow. But here, you can sort of see the lines here. This would be 20 to 30 streaks on that one. The goal of this, I don't know, quadrant method of sector streaking is someplace along the line, you will be putting down individual cells, which will grow up into individual and isolated colonies. And it doesn't matter if it's happening in sector 3, sector 2, or sector 1. It could happen in sector 1 if the stock culture is very low in numbers, that you could streak in this region here individual cells to get individual and isolated colonies. Obviously, that didn't happen here, but you are starting to get isolated colonies grown in sector 2 here. I'll blow that up a little if I can. It might not be. Yes, I can. Yeah, you can start seeing isolated colonies right there. That one would actually be a good one, if assuming that's one. The others are really touching something, so the others aren't so good, but that one looks good. That one looks good. That one looks good. So does that one. So it doesn't matter where your isolated cells will be put. Could be in sector 3, could be in sector 2, could be in sector 1. You just want isolated colonies, and then you pull from one of these isolated colonies, send it on a second plate, streak it out for colony isolation, and then on that second plate, you pick from one colony that's isolated to set up your stock culture. Any question about any of that? All right, remember when we're talking about microbial growth, we're referring to an increase in the number of the cells, not an increase in the cell size. There are three ways that bacteria can grow. Most bacteria grow by binary fission, where one cell simply splits down the middle into two. But there are a few cells that grow by budding, meaning a few species that grow by budding, where a small oak growth, a bud appears in one end of the cell, and then that bud enlarges, forms its own cell, and it will separate from its parent cell when it's almost the same size as the parent cell. I'll show a little video on budding in just a minute. There are also a few species of bacteria that grow by reproductive spores. The spores are tips of filaments at the end of the cell that usually scatter from either like an animal walking by or from the wind, and then they go elsewhere to grow. So you can have a parent cell and it will actually put itself up like that. And that's the media there. And it will make reproductive spores in this region. See if I can draw four of them. The spores are usually at the tip or end of the cell, and that makes it more likely that wind or some animal walking by will pick up a spore and then drop it elsewhere. Come on. So that spore can germinate and then form a vegetative cell, which will then make other cells. Any question about any of that? Reproductive spores is reproduction. An endospore is not reproduction because an endospore, you get one endospore per cell, and then the parent cell dies. So you had one cell to begin with, and then you end up with one cell. That is not reproduction. Endospore is a survival strategy. A reproductive spore is reproduction. We started with one cell, the parent cell. Come on. The parent cell then made reproductive spores, making four of them. Now we have five cells where we started from one cell. So that is reproduction. Any question about any of that? All right, let me show you a little video of yeast budding, similar to bacteria, but it's easier to view with yeast. So we'll talk about it with yeast, and the bacteria follow the same example. Someplace I've got a little video on that, but I don't know where it is at the moment. So let's watch that cell there. That cell's going to make a bud right here. And there it's starting to form the bud. There's the bud. Oops, get out of there. Right there. You can actually see the DNA start to move into the cell. Let me back that up a little bit. So something around here. Right there, the DNA is starting to move into the bud. And that bud enlarges. And then enlarges. And then when the bud gets about the same size as the parent, just a little smaller, you can notice the bud is now separated from the parent cell. And now it's on its own. No longer, the cytoplasm is no longer connected. And then, oops, this cell here can actually start forming a bud. It'll form it here, and that cell will form another bud starting to form here. There you can see the bud starting to form there. Not quite yet, but Around there, that's where the bud's starting to form. And there it's starting to form there too. Any questions about any of that? Oops, let's go through binary fission. Binary fission is simply when one cell divides into two. And what you have is the DNA first will replicate, then one copy of DNA will move towards this end of the cell, the other copy will move towards that end of the cell, and then the cell literally starts splitting in two down the middle, where the cell membrane and the cell wall will be put in place between the two nuclei, which you can see it there. And there we have an actual electron microscopic image of a bacteria starting to divide, partially formed cell wall and cell membrane right there. And then that will fully go across, dividing the cell into two. And you notice once that cell membrane and cell wall is finished, it will literally divide each cell into two. And then these cells can separate and then go through their own reproduction, meaning the DNA will reproduce, and then the cell will divide down the middle again in binary fission. Most bacteria do reproduce by binary fission. Only a few reproduce by budding, and only a few from reproductive spores. All right, any questions about anything so far? Let's talk about the generation time. The generation time is the time required for one cell to divide into two, and it's also the same time as the population numbers to double, meaning a population of 1,000 to double to a population of 2,000 will be the same time as it takes one member of the species to double from one cell to become two cells. The generation time varies considerably between species. It can be as fast as 20 minutes, like E. coli can have a generation time of 20 minutes, but most bacteria have a generation time between 1 and 3 hours. Microbacterium leprae is one of the slowest bacteria cells to grow. Its generation time takes 10 days for one cell to divide into two cells. Any question about any of that? The mycobacterium generally have a slower generation time than many other bacteria, or most other bacteria, because the mycobacterium have a lot of mycolic acid in their cell wall, and it takes a cell a considerable amount of time to make all of that mycolic acid and then put it in the cell wall. Any questions about any of that? All right. The generation time is said to be 20 minutes, but in reality, it should be said to be 20 minutes per generation. So E. coli has a generation time of 20 minutes per generation. So the generation is a time per generation. It's not I don't know, like a miles per hour is how many miles you travel per hour. So don't get that confused. Generation time is the number or how long it takes, how many minutes it takes for the population to double, or the generation to move from one generation to the next. So it's time per generation. Any question about any of that? Students frequently get the generation time incorrect, probably because we are so used to thinking about, oh, I've got to drive someplace, I'll go 60 miles per hour and then get there in an hour. Meaning we're so used to talking about distance or something per hour. And the generation time is the time per generation. Any question about any of that? When microbiologists are graphing the generation time, they like to graph it on a semi-log plot. One shown here. Why is the bottom not showing? There we go. It is semi-log, not log-log, meaning the x-axis is an arithmetic scale where we have the number of generations. 5, 10, 15. So normal arithmetic scale, meaning 1, 2, 3, 4, 5, etc. But then we plot the numbers of cell on the y-axis on a log plot. The reason why microbiologists like it is if they use a log plot, they get a very simple equation for the generation time. Y equals mx plus b is a simple mathematical relation to explain this curve. And if you plot it versus the number of cells on the arithmetic scale, and then the generations on the arithmetic scale, you don't get a simple mathematical formula. What you do is you get a graph like this, which is something like y equals nx plus b plus mx squared plus, I don't know, c, might be another complication in there, and that is a exponential curve. Another reason that microbiologists like putting it on a semi-log plot is that if you use an arithmetic plot, you have most of the points down here. You got 1, 2, 4, 8, 16, all down here. If you use a log scale, the points which are not shown will be scattered more evenly. And then if you wanted to put the next plot on the graph for the semi-log plot, you can put it essentially about right here. So the next point can actually be added to the graph. You may need to extend the graph, but you can actually add it to the graph. On the arithmetic plot, the next point here will be on the ceiling because it's an exponential scale. So you cannot put the next point on the graph. Any questions about why microbiologists like to use a semi-log plot when we're talking about the generation time? And note that this is semi-log because the generations is using an arithmetic scale. It's the number of cells which is using a log plot. When we're talking about any type of organisms growing, they go through different phases of growth, including bacteria. And here I'm going to talk about a bacterial growth curve where there's a lag to begin with, then the bacteria grow very rapidly, then there's a stationary phase, and then the number of bacteria cells decreases. This is seen in almost all living organisms. Like somebody on the East Coast introduced deer to an island off the East Coast. The deer initially got on the island. They didn't start reproducing right away because it was a new environment. They wanted to explore, get ready. And then after learning the new terrain, they started reproducing. And it was a log or exponential growth phase. And then the population of deer entered a stationary phase where they did not grow any further. What happened was the deer had used up or began using up the resources on the island. And so some deer got hungry and died. And then the lucky deer that happened to find food were actually still reproducing, maybe at a slower rate than down here, but they were still reproducing. And that kept the population number of deer about the same. But as more and more time went on, more and more of the resources from the island started disappearing, and then the number of deer declined as there was less and less food for the deer to eat on the island. All right, so when we put bacteria into a culture, let's say you start from a solid media and then you start growing it in a liquid media, the bacteria will enter a lag phase. And that's because you've switched media. The bacteria were used to growing on the previous media. They're not used to growing on the new media. And so the microbes will start making the enzymes and other molecules they need to begin growing. And then, once they have those molecules made, the bacteria will start growing, called the exponential or the log growth phase, where the bacteria are growing initially very rapidly. Why? Because we're in a new environment. There's essentially no toxic substances around, no waste products around, so the microbes can grow. And there's lots of nutrients around and lots of space around, so the microbes can grow very rapidly. However, with time, the microbes will start slowing their growth. And that's because waste products are now around. There's less nutrients around or food. We're talking about an animal. And the microbes can also start deteriorating their environment. All of these will start slowing the growth of the microbes. Why it starts heading towards a stationary phase, meaning the microbes aren't climbing at the same rate they were. They're climbing at a slower rate, if they're climbing at all. When the microbes enter the stationary phase, there are some microbes that are unlucky. They have lots of waste products around, very few nutrients around, and so they're not reproducing. Some of them are probably even dying. But there are some lucky microbes that have lots of nutrients around, very few waste products around, some space around, and these microbes will reproduce. In the stationary phase, some microbes are dying, but other microbes are being formed, meaning reproduction, and the two equal, while the population number stays stationary. That will be followed by the decline or death phase, where more and more of the organisms begin to die. Late in the death phase, the microbes are only sitting around waiting for a favorable environment, or they're cooked and then the microbes won't grow on it, assuming you put it in the refrigerator. Well, why the death phase happens is late in the death phase, there's essentially no nutrients around, lots of waste products around. The environment may be decayed, like too acidic for the microbes. And so there's only cells that are dying or maybe staying stationary. Earlier in the death phase, you do have some cells growing, but there's fewer cells growing than cells that are dying. And that's why we say it has entered the death or decline phase. So there are four phases of growth: the lag phase first, the log or exponential growth next, the stationary phase third, and the death or logarithmic decline phase is last. Any questions about bacterial growth curves? All right, so let's talk about the measurement of microbial growth. There are a number of methods used to measure the growth of microbial populations. There are direct methods and indirect methods. I've used all of these methods myself, except for the most probable number method. You can have a plate count, and that's a direct method. You can have a filtration. You can have the most probable number, and you can have a direct microscopic count, where somebody goes in and starts counting the cells to determine how much the microbes have grown. There are several indirect methods too, and these indirect methods only give you an estimate. We don't really know the exact population of the microbes, but we can get an estimate by using turbidity or metabolic activity or the dry weight. So let's talk about each of these in depth. A plate count is the most common method used in microbiology to get the number of microbes. It measures the number of viable cells. It does not measure the dead cells, because a dead cell will not grow and form a colony. In the plate count, you're actually going to count the number of colonies to get your estimate of the number of cells in the original solution. We call it a plate count, but it's frequently called a viable plate count because you're only counting the viable cells, the ones capable of growing into a colony. It does take 24 or more hours for the colonies to form, so that's one disadvantage of this way. And the results are often reported as colony forming units per mil, not the number of cells per mil, because a colony may form from 2, 3, or even as large as a clump of cells. And that's why we use a plate count. Let's take a look at a little more plate count. First, you make a serial dilution of the original specimen, your original sample, and then you inoculate agar plates from the serial dilution. Count the number of colonies on the plate. You want a count to be between 25 and 250 colonies. Why we do that is to reduce the error in our measurement, and I'll explain that in a little bit. And then you count the colonies on the plate to get an estimate of the bacteria in the original sample. So you make a serial dilution like this, where you have our original inoculum, and then you make a dilution, put 1 ml of the original inoculum into 9 ml. That's a 1 in 10 dilution. You then mix that up, take 1 ml out of this dilution, put it into 9 ml of fresh media. We now have another 1 to 10 dilution, but the total dilution will be 1 to 1,000 because the dilution is the number here times the number there, and that would be 1 to 100 dilution. And then you mix that up, take 1 ml out of that, put it into 9 ml here, and then we have the 1 to 1,000 dilution. Mix that up, take 1 ml out of here, put it into 9 ml there. There we have 1 to 10,000 dilution. And then you can do the same here, put it into here, and there we have 1 cell in that should be 100,000, not a million.
That is 100,000.
Yeah, that's why I said it's 100,000, not. Oh, you're right. No, you're right. That is. I think it was a million. So that is 100,000. I wasn't seeing it right. Sorry. Anyways, you then plate from each of these dilutions an aliquot onto the plate and then spread it around. You can use the pour plate where you usually put 1 ml on and then put that 1 ml, spread it all over the plate. Or you can put 1/10 of a ml on, and then you have to spread it on with the spreader, and then incubate the plates, and then count them. Some of the dilutions will have too many cells to count, in which case you just say TNT, too numerous to TNTC, too numerous to count. The 1 in 10 dilution is that way. The 1 to 100 dilution. The 1 to 1,000 dilution is also too numerous to count, although you can count some of them here. You can't get an accurate count. The 1 to 10,000 is here. You can count that one. And the 1 to 100,000 is here. When you're doing these counts, you want to do a count between something like 30 colonies on the plate to 300. You don't want to do less than 30, and I'll show you why. I guess I could do it here. So the dilution, blow that up. We're going to say 1 to 10, 1 to 100. Oop. 1 to 1,000. You're getting the point here, I assume. I'll do one more, 1 to 10,000. And then you do your count. Let's say this one is 3. This one is 30. And these are too numerous to count. Why you use this one to get your estimate for the original solution is if we were to use this one, our error in the mean, for here, will be 3 plus or minus, whoop, I'll get the plus in a minute, minus 1, which equals the mean plus or minus 33%, 33.3%. Well, let's move that a minus. And that's just from one colony different, plus or minus 33%. Here, our mean is 30, and then let's say plus or minus 1. See if I got that there. 2. Oops. There we go. Equals the mean plus or minus 'I think it's about 3.3%. Whoop, I still have that underline. Which is a much smaller number than this one here. Any question about any of that? And that's why we always use the count that has something like 25 to 250 or 30 to 300. We don't take something less than that because the mean will be have too large of an error. Any questions about any of that? Anyways, that's the viable plate count. The filtration method is similar to the viable plate count, but what you're doing here is you're using the filtration number when the bacteria numbers are very low. So, for example, if we're sampling the bacteria in the drinking water in the city of Vancouver, it's going to be something, but if we were to make a dilution, you're going to get essentially zero on all of these plates because we're doing a 1 to 10 dilution. You might get a few on this plate here, but these others will have zero on them. And that's because the drinking water will have very few microbes, at least bacteria, in it. And what you want to do to get an estimate of the numbers, like in the drinking water, which is low numbers of bacteria, is you pass a given volume through a filter, trapping the cells on the filter, and then putting the filter on an agar plate, and then allowing the cells on the filter to grow and form colonies. And then you can count the number of colonies to get your estimate of the number of cells from your original sample. And remember, that would be not from like 1 ml right here, but that would be something like from 1 gallon. Any question about any of that? There's also a direct microscopic count where a measured volume of liquid placed with organisms in it is put on a gridded slide, and then the cells are counted microscopically. You have to use a special slide. Let me blow the slide up, as shown here, which has a specific volume underneath it, as well as a grid. And usually the slide is designed so if you were to count one square here, this would be 1 microliter of liquid under this square here. And then you would take this number here times a million to get the number of cells in the original solution. So here we have a volume of a microliter, which would be 1 times 10 to the 6. And then you get your count here, that's something like 20 cells. And then your original sample would be something like 20 million cells per mil. Any question about any of that? Now, a bad thing about the direct count, I mean, it does give you a direct answer, but of course, it's hard on the person who has to have hard on their eyes to have to do the count. But another thing is that cell alive or dead? Can anyone tell? Or how about that cell? It's impossible to distinguish a viable cell from a dead cell under the microscope. So you're going to be counting both viable and non-viable cells. And that's true of most of the methods I'm going to be discussing, that they have trouble distinguishing between viable cells and non-viable cells. We're not going to talk about the most probable number method, but let me talk about the indirect methods now. Turbidity has the growth in a liquid culture, and as the microbes grow, the media will become turbid, meaning cloudy. That will let less light through as the light bumps into the cells. You compare that to the amount of light coming through the media itself to the amount of light coming through with the cells here. The number of greater the bacteria, the less light will come through. And you just look to see how much light is coming through and compare it to a chart to get the number of cells per mil causing this much turbidity. Now, it is true, initially you have to set up the table, and then you'd make different dilutions of your cells. comparing it to the regular media, 100% transmission, meaning zero cells, 10% transmission, meaning so many cells per mil, and this one being most of the light being blocked, a much higher percentage of meaning much lower percentage of light coming through, meaning more cells to find your your cell number by turbidity. Like I said, you determine the amount of light getting through, look it up on a chart, and initially you have to make the chart where you do something like 0 cells per mil, 10 cells per mil, 100 cells per mil, 1,000 cells per mil to get your table set up. But once you have the table set up, you can then use that table indefinitely, or as long as the table survives or you survive. And you use that to get your estimate of the number of cells per mil. An indirect method, but it's actually very quick and very easy to do once you have the table set up. So that's why I like doing that one. You can also measure metabolic activity, assuming that the more bacteria are present, the more metabolic activity you'll see, such as CO2 accumulations, and you'll get more CO2 if you have more cells or bacteria present. You may have done metabolic activity in your preliminary biology course. Then the last weight we're going to talk about is getting the bacteria numbers from the dry weight. What you do is you grow up your bacteria in a liquid culture. You then centrifuge down the cells, pour off the supernatant, keep the cells, and then dry the cells, and then weigh them. The more weight you have, the more cells you have. Initially, you have to make a table to determine the amount of weight, the number of cells, and what you'll do is you'll count how many cells you have and measure them. So you do have to make the table, but once you have the table you set up, you can then use the table for the rest of the table's life or your life. One disadvantage of the dry weight method is, once again, you cannot distinguish between the weight of a dead cell and the weight of a living cell. One advantage of the dry weight method is you can, pour off the supernatant and then dry out the cells, but you don't need to weigh it right away. You can then weigh it at your leisure when you have time to weigh it. All right, any questions about this chapter? If not, we're actually quitting 1 minute ahead of time. Any questions at all? If not, I'll see you at 6:30 in the lab if you're in the second lab. All right, bye.